So thank you so much, and thank you for allowing me to present my this talk remotely. I'm sorry that I am not able to join the meeting in person. Today I'm going to present some of the ongoing works that my team and I are working on to explore the connection between seasonal fish spawning and oxygen temperature through the use of an index called our Growth Index. It is still an ongoing work, and I'm going to present to you some of the preliminary results. So I welcome any suggestions or comments that would help us with continue with this study. So this is a simple schematic diagram indicating some of the main processes and components of fish reproduction and reproductive cycle. This includes spawning, production of egg, and egg survive to larvae, and then those continue to grow to recruit back to the populations and ultimately return to become spawners. Specifically, the characteristic of spawning, including frequency, durations, would affect the quality and the quantity of eggs being produced, and that's indirectly affects recruitment, which is an important component of fish population dynamics. Thus, understanding spawning is one of the important components in knowing how fish populations would do, particularly as the environmental condition changes under climate change. Because we know that environmental conditions that fish live in, including the physical and biological conditions, are being changed by the changing climate, global warming, ocean deoxygenation, ocean acidification, and these changes are affecting different processes of the whole reproductive cycle, including fish spawning. Oops. Recently, there are some works already done by Daniel and Lin, one of Daniel's colleagues, that tried to extend the growth principle of growth. It developed a model that help to predict the seasonality of fish spawning. This is a sine wave model that they developed to predict the accelerated the spring spawning of fish and how they may be affected and accelerated as water temperature changes. So in this case, for example, with warming, it will shift the amplitude and the frequency of the model of the sine wave model, and thus with that they can use these models to predict the shift in, for example, the number of days in spawning as temperature changes to a certain degree of temperature. And in this study, we're going to further extend this concept by going into a little bit in details about the different processes and factors that may affect the seasonality of spawning, and thus hoping to explore the use of these factors in predict as spawning seasonality. So this is a really simple schematic diagram looking at a annual fish life cycle, particularly looking at fish spending time in feeding that allows them to grow and develop. And as Daniel just explained, if the conditions is suitable with sufficient food and growth that allow them to reproduce successfully and spawn, and then they will repeat that by continue with feedings, and that go into the next cycle. And this expressed also through the from the metabolic point of view, where in each of these stages, fish would require the metabolisms for covering the routine, and in addition for growth and reproduction. And we know that, for example, during reproduction, it is also a energy oxygen energy consuming process, and that's one would expect that there's also differences in the aerobic scope, where the fish continue with this annual cycle from feeding, spawning, and then back to feeding. So with that, we look at when we look at the literature, there are various factors that may relate to the seasonality of spawning that is also linked to these different processes in relation to the annual life cycle of the fish. There's, for example, temperature and oxygen that affects the aerobic scope of the fish. That's one factor that would affect the seasonality of fish spawning. And then there are also the availability of food. And also some fish are known to also respond to environmental cues, such as photoperiod, that will stimulate them to spawn. In these studies, we want to explore these different hypotheses in explaining the observed fish seasonality, and these are some of the hypotheses that we formulated. So the main hypothesis that we look at is that fish would spawn after period of high food availability, because as Daniel explained, fish needs to feed and to become in the conditions that they can spawn, and also because fish, the metabolism is oxygen limited, so they would need sufficient aerobic scope in order to spawn as well. In this case, if we look at the aerobic scope or the availability of the potential use of energy, oxygen to support growth and reproduction, that would be the period when the aerobic scope is high. And then there are also alternative hypotheses that may explain the seasonality of fish spawning, including temperature, when temperature is high or low, or food availability, or photoperiod. And then we also put in our hypothesis that fish spawn at random, that it doesn't respond to any of these processes or environmental cues. For aerobic scope, we use one of the indicator that my peer students and now people start working with me, Taylor Clark, that developed is an index called the Aerobic Scope Index, which is derived from the von Baudelaire growth functions. I won't go into the mathematics of it in detail. So basically, the aerobic growth index is a ratios of oxygen supplied versus the maintenance oxygen demand. And it is parameterized based on some of the von Baudelaire growth parameters, such as length or weight at infinity. We also use biogeography of the species to estimate temperature preference and oxygen threshold. And we make some assumptions about the activation energy for anabolism and catabolism. And ultimately, the index provide a indications of the aerobic scope. So, for example, when AGI is below 1, it means that the maintenance oxygen demand is actually higher than the available oxygen to them, and thus the habitat is considered not suitable for the species. And at the same time, if warming, with ocean warmings, it increase oxygen demand, or the oxygenations that reduce oxygen demand, that both of that will also decrease aerobic growth index. So we try to explore these hypotheses. We undertake a preliminary analysis using a very limited sample, just 12 fish species, fish stocks, just to test out the methods and see and get some initial results to inform us about the next step. And we selected species that are a different life history and geography with some of the small pelagic species, gyroid species, flatfish or halibuts, and some of the other fish species that have quite well-known fish spawning seasonality. And then we use environmental data, and because of the limitations of some of the variables that we get from some regions, so we use historical simulations from Earth system models for oxygen, temperature, and the parameters that's needed to calculate AGI. Also, food availability is indicated by net point predictions. And we use species distributions to inform the exposure of the species or the stock to these different environmental factors. Here I use stocks instead of species. Stocks here is defined by a particular species at a particular exclusive economic zone. Because we know that some wide-ranging species have widely different environmental conditions within their species range. So we want to focus on a specific areas where they share some similarities in environmental conditions. So to test this hypothesis, we use a rank-based, we chose to use a rank-based or non-parametric indicators. So it is basically looking at the month at when some of the indicators are ranked in terms of their values of high or low. So, for example, for the first hypothesis, where the fish spawning is related to aerobic scope and net point predictions as indications of food abundance, we look at the overall indicator is a additions of the ranking for the aerobic scope and the MPP. So whether a month has both high or AGI or MPP, then it will get a high value of these particular rank-based indicators. And similarly, we use a similar approach to look at temperature, only net point prediction only. We also look at photo period. And then we use a random selections of spawning month to test the as a random as a null hypothesis for us to compare the performance of other hypotheses. And we use the data from FishBase. So we look at we go to FishBase, obtain the seasonal spawning informations, and then use that as the variables to test the hypothesis. So this is the results of this preliminary analysis. The index basically is the rank-based index for the different hypotheses on the y-axis, while the x-axis is month of a year, and this is the climatology from 1970 to 2015. The gray area is the spawning season. So if you see from these two examples for cod and for round herring in the Gulf of Maine and Senegal respectively, the hypothesis ones are main hypothesis where the spawning is related to the aerobic scope and food is really closely related to the spawning season. And secondly, or the next best is the photo period, where particularly low photo period seems to match up well with the known spawning season. And temperature or net point predictions individually doesn't match up as well as these two other hypotheses. So we repeat this text for all the 12 species and then compare them quantitatively. We use a number of test statistics and metrics to examine the performance of these different hypotheses. Calculate the accuracy index for predicting known spawning months based on the index. We also look at the precisions or the area under curve as indicators. The higher the values, the better is the performance. So in this case, like the two examples that I showed across all the 12 species, the main hypothesis of aerobic growth and food availability, and secondly, the photo period, particularly the short photo periods, perform best, and both of them are very significantly, largely significantly, highly significantly different from the null model. And while the other hypotheses are considerably less accurate and precise in predicting the month of spawning. So this is kind of encouraging. It shows that our main hypothesis that we put forward, that both aerobic growth and the food availability seems to be the main factors affecting fish spawning. And then, as we know, numerous fish species respond to photo period in terms of the spawning time. So it also confirmed that aspect that is looked at in the published literature. So this is just a preliminary result, and we think that some of the extensions with this, if we can really establish this further, this could be extended to further understand spawning and the impacts of climate change on reproduction. So, for example, we may be able to identify species that have a fixed queue, such as photo period, or flexible queue that is dependent on environmental conditions, oxygen, temperature, and net point productions as different types of spawners. And also, one of the mechanisms of climate change impacts on reproductions is through reductions of the quality and the quantity of eggs, or the increased likelihood of skipped spawning. Some of a topic that was just discussed earlier in the discussion sessions after Daniel's talk. So that's very interesting. For example, for fixed queue spawners, if there is a mismatch between the fixed queue, such as photo period, and the optimum temperature, oxygens, and food conditions, it may result in the reductions in the quality of eggs, or as Daniel explained, it may also result to skip spawning. And then for flexible queue spawners, it may actually change the spawning time or the quality of the optimal conditions that may ultimately affect also egg quality and egg quantity. This is my final slide. The next step is to test hypothesis using a much bigger data set. FishBase has more than 3,000 species records of fish spawning. So I think that provides really rich resources for us to test to explore further, and then if this can be established, we want to then apply these methods to examine how the changing ocean conditions, changing temperature, oxygens, and net point predictions will affect fish spawning. We also want to compare and contrast with the other published models, for example, the one that Daniel and Ling published, and I know Daniel is really interested in this, and also examine the implications of changes in spawning, the bigger implications on fish biomass, catch potential, and the implication for fisheries management. So with that, thank you very much.